Good afternoon, everyone, and a warm welcome to our Fellows Public Talk series. I'm Claudia Rizzini, the Executive Director of the Radcliffe Fellowship Program at Harvard University. It is my pleasure to introduce Anne M. Robertson, Professor of Mechanical Engineering and Material Science at the University of Pittsburgh. In the United States, an estimated 2 to 8% of the adult population have unruptured brain aneurysms. Many of these aneurysms weaken spots in the walls of the cerebral arteries that bulge and swell over time will go undetected. When aneurysms do rupture, however, the consequences are devastating. They carry a 50% fatality rate, and survivors often suffer significant neurological damage. Certain populations are disproportionately more likely to develop brain aneurysm, including African Americans, Latinos, and women, especially women over the age of 55. Anne Robertson, a mechanical engineer with a background in solid and fluid dynamics, approaches biomedical problems like brain aneurysm from a structural point of view. Her highly interdisciplinary approach integrates principles of fluid and solid mechanics, computational modeling, and advanced imaging techniques to understand the underlying mechanism of disease and tissue behavior. As co-director of 2 NIH-supported multi-institutional R01 programs, she applies these approaches to brain aneurysm, as well as the development of a digital twin for the bladder. While at Radcliffe, Anne has been continuing to look at different factors that affect the strength of the wall of unruptured aneurysms, including the role of calcification and the vasa vasorum, small blood vessels that supply the aneurysm wall. She hopes her work will continue to enhance the diagnosis and treatment of disease of the brain arteries, ultimately improving patients' outcomes. The Q&A portion of the public talk today will be moderated by Li-Shin Sun, Hardy Fellow at the Radcliffe Institute and Professor of Biological Sciences at Central Washington University. And now please join me in welcoming Anne M. Robertson.
Okay, it's my great pleasure to be here and to talk to you a little bit about some of my work. Before I do, I just want to thank my fellow Radcliffe Fellows for your friendship and inspiration. And during your presentations, each of you have brought something and shared something from your own world. And I've been deeply impressed by your devotion to pursuing a topic relentlessly and deeply, often simply because it fascinates you. And this is such a talented, impactful group, and I still remain a bit in awe of each of you, awed by your success in pushing your work to the very highest levels amidst challenges and continuous competing demands on time. I'm really grateful that I've shared this journey with you. So today, I'd like to bring you into my world, the world of an engineer who's looking at a biomedical problem. So thank you for being here. So what I'm going to do today is talk a little bit about soft biological tissues, give you some background, and address a long-standing conjecture on what makes soft tissue soft. Then talk about cerebral aneurysms, which is a special type of biological soft tissue that Claudia mentioned, and then turn our attention to vasa vasorum. So first, I'd like to just talk to you qualitatively about what is a soft tissue. If you look at your hand and the skin on your hand, you can pull it up, and it seems to give a little bit, but then it sort of locks up and stops moving. This is a soft tissue, and we have many other soft-like tissues, and you can contrast them with bone throughout our body. So the work I'm going to be talking about today has many applications to other tissues as well. The focus of my research has been on blood vessels as soft tissues, as well as the bladder, as Claudia mentioned. So today what I'd like to talk to you about is largely the cerebral aneurysms, but before I do, I just want to mention another area of research that I've worked on that inspires some of my work on aneurysms, because we might think of aneurysms as something terrible that's just about to rupture, but many of them have actually remodeled themselves and are quite strong and stable. Some of my earlier work has been with Yadong Wang, who's now at Cornell University, on in vivo tissue engineering. And this is really cool work where he developed a scaffold, a cylindrical scaffold without any cells in it, that could be put in place of a section of artery into a rat model. And as blood courses through this, cells move into this wall and start manufacturing matrix and start to build a blood vessel while that matrix dissolves away. So the body has amazing capabilities for reconstructing things and healing things, as many of us know. So as Claudia mentioned, my group is really interested in how these things happen, what structurally is going on to give the blood vessels and things their properties. And we use experimental techniques, mechanical testing, as well as bioimaging, computer modeling, and mathematical modeling to try to understand this process. Before I go into the research aspects, I just want to get everyone a little bit of background, because I really want everyone in the room to understand what I'm talking about. I'm really excited to share it with you. So first of all, a key concept is force versus stress. You can think of force, you're pushing on a table or something. That's the overall force you're applying. But if you break it down, it's per unit area, it's really the stress. So that has a different effect. If your fingers are pushing on it or your whole hand is pushing on it, the stress will be different. So in particular, for this application, stress is more important than force for failure. So here, in the theme of cats, which is permeated through our group at times, we'll start with the cats. So if you imagine a cat walking across the snow, here this poor cat has fallen through the snow, but if we add snowshoes, this cat can now happily walk across the snow. What is different? The force is not different. It has the same weight. But the area that force is distributed over differs. In one case, area 1 is smaller than area 2. And as a result, the stress, or weight per area, is smaller in case 2, where the area is larger, where the snowshoes are being used. And as a result, that force has been distributed over a large enough area that it doesn't break through the snow. It's less than the strength. So this is a little bit of key thing to think about. Stress is important. So what are soft biological tissues? Besides our idea of skin, we can be a little bit more quantitative about that. So I'm going to give you a sort of idea of an experiment, and this is how things used to be experimented on, but now we have fancy instrument machines and things to do this in a more sophisticated manner. But imagine we wanted to run this experiment, and we took an artery and hung a force on it. It would stretch a certain length. Stiffer things would stretch less than, or move less down than things which are more flexible. But we could take some material like an artery and apply different forces to it, and as a result, it would have different stresses, and we could see how much, we could measure how much it extended or changed its length, and we could normalize that by length. And what we would find with soft tissues like skin is that we have this initial region which is very low stiffness. It gives a lot with very little additional load. That's why the slope is low, because it can strain a lot with very little increase in stress. On the other hand, it then kicks in, something happens, and we're going to investigate what that something is, where it then becomes stiff as we get to higher and higher lengths. So this is qualitatively and quantitatively what soft tissues are like. So Wertheim in 1847 seems to be the first person to notice this qualitative difference, this J-shaped curve, as he called it, in arteries and other soft tissues like veins and tendons and ligaments that are the source of this qualitative difference. They all have the same general shape. So why is this important? How do blood vessels generate such a response? And how is this changed by disease? And how can we use this in medicine? So these are some of the questions that we are asking and curious about. Why is this important? I think you can all imagine why. We want the skin to give a little bit as we move around, but we don't want it to overextend, to extend too much. In fact, there's genetic illnesses where it will extend too much, and usually it's more fragile then as well. So we want it to kind of lock in at a certain point. So this is very useful for arteries, for bladder, for many other tissues in addition to skin. Then comes the question of how do these soft tissues generate such a response? So first, in order to understand what's going on to be able to have this response, I'd like to talk a little bit about the main building blocks. So the main building blocks, we have collagen fibers, and you can think of these as kind of cords. They're a little bit flexible, but not very flexible. And then we have elastin, which can be in the form of fibers or as sheets. And this is very, very flexible. So think of like a balloon. So how can we combine a balloon with stiff cords to make this J-shaped curve? That's the question we're going to get at in a moment. In addition, we're going to come back to this. This soft biological tissue has cells in it. And these cells are living things, and they can do amazing things. They can sense things. So for example, this is a picture of an artery, and we can see these cells lining the interior of it. Those are endothelial cells, and they're there, and they can sense shear stress on them. So they can, in a sense flow rate. And as a result, they can do things, respond differently if it goes up too much. We know, for example, if the shear stress goes way up, if the flow rate goes way up, they will actually release chemicals and cause the surrounding smooth muscle cells to relax more, and the blood vessel will get bigger and return the shear stress to the homeostatic level. So these are things they can do. They can contract and they can manufacture things such as these acellular materials. So they're important for the survival of the blood vessel and soft tissues. So how do we achieve this J-shaped curve? Burton had conjectured initially that this curve was due to the fact that elastin and collagen were, sure, they're working in tandem, but the collagen didn't really start load-bearing till later. So the initial part of the load-bearing was due to the elastin fibers. So here we can see the elastin fibers. We start pulling them down. They're wavy at first. Then eventually they get straight and they start load-bearing, and we pull further and further and further, and now we have this curve over here for elastin. Collagen, on the other hand, is much stiffer. We know that. But it's also wavier. It has higher undulation, so it kicks in even later at a larger length. And as a result, when we combine the two, we have this nice J-shaped curve. So that's what Burton conjectured. And you might wonder, why is this so important? It's very important because this flexibility that we talked about for skin is also very important for the function of artificial blood vessels. You know, how do blood vessels change with age? Have they lost these properties, et cetera? So we'd like to prove this conjecture. The challenge with this is we would really like to be able to see into the blood vessel and see these things going on with the fibers while we're loading them and making these measurements. So we have some options for microscopy. We can use probably what you used in high school, which is two-dimensional classical histology. You have little thin slices of tissue on a slide. You put it under the microscope, put stains on it, and you can see all sorts of parts of the cell wall. Fortunately, things have changed, at least since I was in high school, and Doug was in high school, and my high school classmate is here. So we can also now take advantage of the fact that there's optical methods that enable us to image planes, stacks of planes of the tissue, so we can put a whole artery in there and just image at different depths and then recreate in 3D this object after staining it. So we can do that with confocal microscopy as well as multiphoton microscopy. I was very, very fortunate that we have this fantastic imaging facility run by Simon Watkins at the University of Pittsburgh. And he was one of the first to get this multiphoton microscope. It is amazing in the sense that you can actually image both elastin and collagen without cutting up the tissue, without fixing it, without staining it. So you can just take a piece of artery, put it under the microscope, and see all the stuff that you want to see. So being engineers, we're like, wait a second. Now we have a chance to prove this conjecture, because we can build an experimental device that goes under the microscope, and we can image those things while we're stretching it. So that's what we did. We built this uniaxial system that can pull on the tissue under the microscope, and we could see, for example, here we can see these wavy collagen fibers that are from a strip that goes around the artery, like a little circumferential strip. We open it up, pull it, and we see that these wavy fibers become straightened. So now we have this opportunity combining the microscopy with this measurement device to try to explore this conjecture. So as I mentioned, we can get this data from our device. This J-shaped curve came up from these arteries. But we could also look at these different loading levels, what's happening to the fibers. And what we could see is, just as conjectured, as we load and load and load, this waviness is being pulled taut, and eventually the collagen fiber becomes straightened and starts load-bearing. So we were able to quantitatively trace these fibers, build them into constitutive models, have this sort of distribution function of waviness and angle and things like that to mathematically recreate this data set just with these functions and just based on this conjecture. So we proved basically this conjecture that we have this elastin-dominated region, collagen recruitment, and then collagen-dominated. And this, I just want to give a shout out to PhD student Michael Hill, who was the key researcher on that, and at that time, a younger Shin-Jae Dwan, who had just joined the group. So this is important, this connection between the properties inside the wall and its behavior for developing these material models, but enables us to really start to understand, why is this change in aging happening? Can we look at the structure and understand why? Can we try to reverse it? Can we try to make treatments where we've lost that toe region, or we can look at how this affects the failure mechanisms. So this kind of approach has been used now in many, many different biological systems, soft tissues like cornea of the eye, tricuspid valve of the heart, lungs, bladder dysfunction due to spinal cord injury, outlet obstruction like in our group. So it's been quite used. So just a few of the takeaways from this part one. The soft tissue exhibits a J-shaped curve reflecting this unique mechanical behavior. And we've confirmed this long-standing hypothesis about why that occurred. This curve arises from a soft elastin-dominated phase followed by a stiff phase, and these findings enable us to develop mathematical models and understand tissue mechanics for a wide range of tissues, not just the arteries. So now I'd like to turn to cerebral aneurysms, the next part of the talk. Before we talk about the aneurysm itself, I'd like to talk a little bit about the circulation in the brain. This is really cool. So this is actually from a human cadaver. It's a circle of Willis. We have real access to these all the time in my research group, so we can really explore this. So it's nice to ask yourself, like, how does the blood get to our brain? We know we have carotid arteries. The two arteries are feeding the brain. We have two vertebral arteries feeding the brain. One possibility is that each artery would feed a different quadrant. That would seem to be reasonable. But fortunately, that's not what happens. Fortunately, it feeds into a circle and then is redistributed. So if someone was to block off your carotid artery, you still get blood to the circle from the other three vessels, and you still get blood to your brain. So we can see these four vessels in this human circle of Willis. This is a very good one, by the way. A lot of people don't have a full circle, but it still works, so don't worry. And then these yellow arrows are pointing to the vessels that are feeding your brain. So 4 vessels in, but all these vessels are going out, even more that we can't see, but these are just some of them. So all those vessels are feeding the rest of our brain. That's all good news. The bad news is this is also the place where the brain aneurysms tend to form. They tend to form at bifurcations in this circle of Willis, which of course is now a concern. So this is looking from beneath the brain in the same orientation as that circle of Willis, if you want to make the connection. Okay, so like I said, these are typically found at the circle of Willis. They're typically saccular in shape. You can think of them as like if you had a tire and there's a weakened region that bulges out, kind of like Claudia said. The good part is they're actually alive. So in many cases, they're able to reinforce that wall. It's just not just a thin bulge out thing like a tire. Cells can go in there, lay down collagen, and reinforce that. So now it's a bulge, but it's a stable bulge. And that's why there's actually, even though there's 1 in 50, probably someone in this room has an aneurysm somewhere, it's not such a big deal in general because there's a low rupture rate. It's less than 2% per year, and many of them are remodeled like this. So that's the good news. Then this is like not so good news, why we're trying to help again, is the treatments are themselves a little bit dangerous. So one of the treatments is open brain surgery where the neurosurgeon goes in and places a clip across that bulge. So that's kind of like putting a clip across the bulge on the tire to isolate that weaker tissue. The other things are more like putting glue in that bulge on the tire, where you put in coils that can be done endovascularly or a flow diverter to drive flow past that so that a thrombus forms, kind of like putting glue in that little bulge to isolate that thin part of the wall. So those are effective, but they're also risky enough that if a patient comes in, you're not just going to immediately treat them. That patient comes in with an unruptured aneurysm. So there's different ways of assessing, trying to make decisions about whether people should have surgery. One is by the size of the aneurysm. For example, you can see that there's a heavier scoring for much, much larger aneurysms. They're more likely to rupture. One is whether they have these bulges and undulations. One is where they're located. They're more likely to rupture if they're in the back of the brain. So these are factors that are considered. However, there's big challenges with that. One is that almost most of the aneurysms are small. Even though they're less likely to rupture, most of the aneurysms that rupture are small. So that's a big dilemma. Of course, if it's a big aneurysm, you're kind of expecting it might rupture. But if you have a small aneurysm, it's still a concern. So what we would like to do in our group, I'd like to tell you a little bit about this logic. Like, how do we know larger aneurysms are more likely to rupture? The data and the approach to that largely is based on the following. It would be like us looking at all these cars driving down the road and trying to say which one is going to break down. Okay, so all these people are walking around with aneurysms, which one is going to rupture? Currently, the approach is to say, let's look at the broken down cars on the side of the road. Let's see, okay, this one is broken down as a flat tire. Let's go see if we can find flat tires in the driving cars. So we're looking for things in broken down cars to try to guide us in taking cars off the road and having them repaired. So in the case of aneurysms, what this means is, let me show you these two aneurysms, same aneurysm. One is before it ruptured, that's what it looked like, and the other is after rupture. So if we look at these after ruptured cases, or the broken down cars, we might say large volume, lobulations, complex geometry. But if we went back to look at the unruptured case, it would look completely different. We would not have predicted that this was going to rupture in fact, 18 days. So that works up to a point because things change dramatically just before rupture. Just like with your car, the car tire probably looked reasonably fine and then it had a flat. So it's something we have to try to get around. So in our group, what we've been trying to do as mechanical engineers, for the most part, is to complement this association study with causation. Can we create a kind of stress test, which would be like taking your car off the road and running it through tests in the garage, revving the engine, testing different things. So we would like to take the aneurysm tissue and run stress tests on it. Not before, unfortunately we can't do that in vivo, but I'll tell you what our strategy is ex vivo for this. And then we can also use this to identify multiple modalities for failure. Right now, all the failures being more or less categorized together, we might be able to find out, once we can find out, like that one failed because it had a flat tire, we should run a different kind of statistical test with this kind of information, having more information. So what we do, instead of doing it in vivo, which is not possible, is we're fortunate that one of the methods for treating aneurysms, which is quite common, is, as I mentioned, open brain surgery, where you go in and you identify an aneurysm. Here's a reconstruction of 3D DSA data on the left, where you can see this lobulated aneurysm. And then you can see the surgeon operating on it to put a clip on it. Once the clip is put on, that aneurysm wall is secure. And you can clip off the other side of the tissue from the other side of the clip. That's then sent to my laboratory, and we start doing all sorts of tests on it. So this is an example specimen. So this is a major undertaking. We're not just doing it in our lab somewhere in a corner of the building. We're fortunate to collaborate with many, many different groups. So this project that I co-direct with Cerebral, he's a computational scientist. We have been doing this since 2016, actually before that with R21, but we have this sequential RO1s where we're working with 5 hospitals and many talented graduate students that I've listed here, and many talented neurosurgeons to obtain these samples. And we have already acquired 194 samples from these different sites, which I understand is the largest aneurysm bank in the world. But we also acquire 3D imaging data, patient questionnaires that we make up that have to do with lifestyle, for example, about the condition of their gums and teeth, missing teeth, et cetera. Also things having to do with their lifestyle choices, as well as patient data from their charts like BMI, weight, et cetera, age, medicine that they're taking, surgical videos and images, blood panels, and op reports. So the idea then is to have a direct mechanistic approach to understanding aneurysm rupture and stability. So just to give you an idea what we do once we get this tissue. So we get this tissue, we then put it in high-resolution micro-CT, we make a model of it, a computational model. From that computational model, we can get wall thickness, we can find out where it's thin, where it's thick. We also take the data sets from the clinical imaging and reconstruct them into this 3-dimensional vasculature, and we map the model of the tissue back onto that. Because then we can see how the fluid flow is related to changes in the wall, and we can also take that specimen and do bioimaging on it, mechanical testing on it, and help use that data for a solid mechanics model. The idea being that these are other things, like the fluid mechanics could come in as a new biomarker if we understood more how it's affecting the wall. It's believed to be deleterious in general. But we would also like to be able to predict, based on the geometry potentially, if an aneurysm would rupture, and that's our solid mechanics study we can do. So the basic logic then is we're first going to use these stress tests to look at walls that are like gold standard, healthy aneurysm walls, the ones we don't think are going to rupture. And then we're going to try to understand the multiple causes that are probably responsible for failure. And then ideally, we can improve our risk assessment and design new treatments potentially. So let me tell you a little about the stress test. So the stress test that we're doing is to take an aneurysm, which you can see here, it looks kind of like an egg. You're cooking in the stove, sorry about that. But then these are typically quite small. If they're 4 millimeters or bigger, we can mechanically test them. So this is, one of my students currently has developed a cutter for these, which is really wonderful. This one was done by hand. Neurosurgeons trained my graduate students to do the cutting of these things. So we cut these into like a dog bone shape, which is a preferred shape for mechanical testing to failure. And then we run these tests. So what we see here is stress, which we now know stress is force over area, versus stretch. And we can see the basilar artery, which is one of the brain arteries. It has this J-shaped curve that we were talking about. It's very elastic at first, and then it kicks in, it's stiff. And then we see the data from the unruptured aneurysm. So these are people who are walking around, the unruptured aneurysm, what should we do with these people? We can see very interestingly, first of all, that the aneurysms don't have that long extensive region, that soft region. So what component do you think is missing from the wall? That elastin component. And in fact, aneurysms don't have that elastin component. So this is completely logical. Then in addition, with this stress test, we in fact identified two groups of aneurysms. Ones that are weaker, so they will go to a lower maximum stress before failure, because these tests were stopped at failure, and ones that are stronger. They're not as strong as the basilar arteries, but they're strong enough from computational studies of the wall properties, wall behavior. So the question is, can we now look at this group of vulnerable and strong and understand differences, understand why the strong are strong, and understand possibly multiple reasons that the vulnerable group are not strong? And we can see they have different shapes. Some of them have no soft region at all, and some of them do. So maybe those are weak for different reasons. So what we're going to do now is we're going to take a look at two of these cases. One, cerebral aneurysm 25, labeled that, and one, cerebral aneurysm 65. One is weak, one is strong. Let's dive a little bit deeper to see what might be different about them. So we'll start with this strong case, the so-called gold standard aneurysm, one of the ones we would like to have if we have to have an aneurysm. And so what I'm going to show you is this imaging of a small section of the wall. So just to give you an idea what's going on, we can image more of the wall, but I just want to focus your attention on this because it's slightly curved surface, so it's better to concentrate in an area that's at the same vertical plane. So essentially what we're going to do is we're going to look at, like I said, the stacks of images that we can get from multiphoton microscopy, and we're going to start looking at the blood side, and then we're going to go and see in this movie in a minute the outer side. And what I want you to look at is the angle of the fibers. First they started at minus 50, then they shift over to 50, and then they become wavy, and they're also more in multi-directional. Oops, sorry. So this is actually, if we think about, a very good thing. If we were going to, check out possible parachutes we could use to jump out of an airplane, we would not want ones with just single direction fibers. We would want one with two directions, right? Because it's a biaxial load. Single directions would pull apart. That would not be good. We want them to be together. In addition, wouldn't it be nice if there was a second layer of wavy fibers that would kick in once we really heavily loaded the fibers beneath, which is what we see is happening here. So this is a very well-defined collagen structure with very distinct collagen fibers. Let's go see what happened to poor CA65. So this aneurysm wall, we took a look at. We first took it, we analyzed it under micro CT, we created a 3D model of it, but we could also see not only the geometry, but also some of the things inside the wall. What we see here is this yellow. Those are microcalcifications. So we can see this wall is laden with microcalcifications and also some lipid pools as well. So then, I'm showing you a movie that was taken while that data was acquired for CA65. So that we'll see it pull up and we'll see it lose its strength at the top of this video. So it's being pulled, and we can see that what happens is it starts to tear. Even in this, without being under the microscope, we can see that it's starting to tear on the left edge there. And then it's tearing from around this region. And we could go in with, I don't have time to go through all the details, but we could go in and look at this under multiphoton, see how it tore, where it tore, because we fixed it as soon as that happened and went to look at it just so it wouldn't fall apart. And then we also performed computational simulations. My colleague Spandan Maiti, who's a faculty member in bioengineering at Pitt, along with doctoral student Ronald Fortunato, performed these simulations where they included individual fibers in a model, as well as this rigid calcification, and looked at where the stress concentrations formed and found that they formed in these locations where this thing failed. So the takeaway from part 2 is that brain aneurysms are bulges in brain arteries that may rupture. They can be fatal or cause lasting neurological damage. However, many never rupture, and treatment carries its own risk. So to guide treatment decisions, we are using these mechanical tests, stress tests, to distinguish strong versus vulnerable. And then we're now matching this with bioimaging and mechanical testing to understand more about what is the source of that failure. And I don't have time to talk about the numerous other things we've looked at in terms of geometry, blebs, collagen fiber organization, but these are 2 examples, I think, to illustrate it. Okay, so now let's go to the vasa vasorum and brain aneurysms. The vasa vasorum is a Latin words for vessel of the vessels. And so you might wonder, why do blood vessels have vessels? And I think it's more obvious if you think about the heart. The heart is full of blood. So it can get oxygen diffusing from inside into the wall. However, it still needs blood vessels. It still needs coronary vessels. And in fact, when people have heart attacks, it's usually because those have become obstructed. So that's an example of where the diffusion distance is too big, because it's a thick wall of the heart, plus the heart has all these cells inside, smooth muscle cells that are contractile, that are using up the oxygen. So it needs to have another source, all these coronary vessels. Now, the coronary vessels themselves need vasa vasorum, actually, because they need vessels too, as do many other vessels in the body. Typically, it's when they get thicker, the diffusion from the inside is not sufficient to meet the metabolic needs of the cells inside, and so these other vessels will form from the outside. So much of the work in the literature, nearly all of it, is looking at these vasa vasorum using this classical 2D histological cross-sections. So in this image, we're looking at a coronary vessel, cross-section of it. The lumen is in the middle. The adventitia, this outer layer of the wall, is where we can see these tiny vessels that if we zoom in, we can see this is a vessel feeding a vessel. But we can't really tell much about it in a 2D cross-section. So we're going to come back to that in a minute, but that's just to let you know that in general, we think of these vasa vasorum as being a positive thing, and we would like to be able to image them even better if possible. Now, when it comes to brain aneurysms, if you look at the literature on brain aneurysms and vasa vasorum, there's a large consensus thinking that it's negative. And that's because they're associated in these kind of histological studies with inflammatory cells and atherosclerotic type changes, in addition to focal edema, so fluid is leaking out potentially leaking out of these vasa vasorum into the brain tissue and causing swelling, and also aneurysm growth. And as a result, there's proposed treatments to use drugs to actually prevent vasa vasorum from forming, so using inhibitors of angiogenesis. So do we think this is a good idea? Of course, people are going to be researching this more and cover this, but I think we can say, currently we can say there are certainly cases where it seems to make sense. So here's an example of a 57-year-old woman who presented with severe walking problems and also swallowing problems, and was brought in for imaging, and they found out that she had this giant 2-centimeter aneurysm, which is huge. So for a variety of reasons, they decided that the best treatment at that time was to put coils in it. So that's like putting, remember, super glue inside or glue inside of the tire to try to move the wall away from the circulation so it's less likely to break. Okay, that worked initially, but then the symptoms worsened, and in fact, when they did more imaging, the aneurysm had grown. So they were puzzled, how could this have happened? How could it have grown if it's not getting blood? So they decided to put coils also in the feeding blood vessel. Worked at first, unfortunately then it worsened, so they brought the person in and did surgery. It turns out that vasa vasorum had formed and bypassed all these occluded areas. They'd gone and found their own way to get to the wall and start feeding it. They'd found their own way to get to the blood vessel and start feeding it, and so the aneurysm could grow, and this treatment was not successful. So we might say that this would be a good candidate for trying to use things to prevent angiogenesis, if this is your treatment that you desired. But we've been looking at things a little bit differently because we're looking at all these healthy aneurysm walls, all these walls that have restructured themselves and are actually strong and robust. So in those cases, often the wall is getting thicker as part of this strengthening process. So that would definitely need some consideration about whether these vasa vasorum are important in that condition. So before we look at the brain aneurysms, I just want to show you a few images from some of the work we did very recently. In fact, my 2 undergraduate Harvard researchers have been somewhat involved with this, particularly Evelyn, who's sitting there in the back, is looking at these vessels and looking at the vasa vasorum. So Mehdi Remezanpour, a very talented doctoral student, did this imaging, and this is massive imaging. I don't know how many hours. I think we got a little bit of trouble with the CBI for taking so much time imaging all these different blood vessels, but hours and hours and hours to reconstruct multiphoton images of this. So this, Mehdi came up with a clever technique of using colorization based on angle so we can see the vasa vasorum even more clearly in these. So this, remember, is in contrast to how we would use classical histology. This gives us a whole perspective. We can see the 3D nature of this going into the wall, and we can see its orientation. We can count how many there are instead of sectioning things and trying to count them. We may miss a section if we do sectioning. We may damage the tissue. We may get artifacts. So this is a really valuable approach. This is another image of a vessel with a vasa vasorum. We're now seeing multiple colors because we've turned on the green channel, which is for this elastin component. So we can see red is the collagen fibers, green is elastin, and we can see this fiber diving into the wall. It's very interesting because then we can take an orthogonal cut of the 3D stack, and we can literally see this thing diving in. So it's going into the wall and feeding that deeper layer. So let's ask ourselves, what would an effective vasa vasorum look like for a sacral aneurysm? So imagine that you had a garden that you want to put water in. Would you run a whole bunch of individual hoses? Could you come up with a better way of being able to make sure that it gets water? So we can think that it may be different than this idea of single vessels feeding all these different spots. So the next thing that was done, and this Yas Tobe and Muha did a lot of work on this. Abdurrahmanov, sorry, I should ask Elena to help me with the pronunciation of this. We'll talk afterwards. We took 3D images from the clinic of this patient who had 3 aneurysms, in fact, and you can see them labeled here. The clipping, open brain surgery, was used to place a clip on aneurysm 2, and we were able to get tissue from that. So this tissue was sent to our laboratory, and we used a protocol that was developed by Yas Tobe, who I just mentioned here. And he came up with a protocol where we could massively stain this vasa vasorum. So you see here this vast network of blood vessels, these interconnected vessels that are on this specimen. So in this case, the green is the vasa vasorum, and the red is the underlying collagen fibers. And we can see some magenta that's actually us staining for calcification as well. So I want you to take a look. So remember, for the cerebral blood vessel I showed you a moment ago, we saw the blood vessels coming in from the outside. We also see that happening here, but they don't dive down into the middle of the layer. We'll see in a moment. But you can see these colorized vessels here. These are the ones from the outside. So I'm going to show you a video in a moment. So keep an eye out for those blood vessels that are actually coming from the outside. So we can see those here. You can see, for example, this blood vessel here that's coming out and diving in. And then we can see this vast network of vasa vasorum that's actually just below the surface, but it's fluorescing, so it looks like it's on top of the surface. And this is a cross-sectional view where you can see it's right at the surface. So if we contrast this with this brain artery we saw a moment ago, where the brain artery has a single vessel coming in and feeding it, what we see in this case is essentially the idea of watering the garden with a network of hoses that are all permeable, so the water can leak out and cover a larger area effectively. And it's at the very topmost area of the aneurysm wall. And we're still in the process of investigating, why doesn't it need to go deeper if this is a relatively thick wall? So evidence, now we want to say, well, this could be bad still. Just because there's a lot of it doesn't mean it's good. Maybe this is bad. So if it is playing a positive role in bringing oxygen where it's needed, we would expect it to be denser in regions that were thicker, because those regions need more vasa vasorum, just like there might be regions of your garden that needed more dense watering. So in this picture, what we're seeing is wall thickness is identified by these different colors. The thickest is red, and the thinnest is this green color. And we can visually kind of see that the vasa vasorum, which is overlaid on this in gray for this image, is denser in that region. But we can also quantify that. We can, from the projection on the surface, we can figure out what is the areal density on that surface, and then map that as a function of these different regions of wall thickness and see that it's, in fact, most dense in the regions that are thickest. So this is consistent with the positive role of vasa vasorum in supplementing transport to thicker wall regions. The other signs of a positive influence could be to look at the wall structure. So remember, we're concerned that potentially this vasa vasorum are bringing in inflammatory cells, which would tend to break down the collagen fibers. So we can look at the wall and say, hey, are they broken down? Is this degenerated? So in prior work, we had come up with collagen categories, some being characterized by diffused imaging under multiphoton. We're now still exploring this under other imaging modalities to see what is really going on there, but we believe these are degenerated collagen fibers or new collagen fabric that hasn't formed distinct fibers. Then we have fibers which are distinct in their uniaxial. There may be conditions where that is a reasonable thing to have a uniaxial direction, as well as multiaxial. So in this specimen, I'm now just showing just the collagen channel. You can sort of see it, but I'm going to give a zoomed in region here. Yeah, you can see the zoomed in region just to get a closer look. And we can see that they're multidirectional, just like we were talking about would make sense. So our conclusion here is that this collagen fabric is also consistent with a positive role. We don't see degenerated fabric like we might see if there was inflammatory cells in there. The most recent thing we're doing, and this has all been work that's been done during this sabbatical year, is to look at the flow and see if we can, from modeling, look at if there's sufficient oxygen transport, if the transport is also modeled. This is work that's been done in Juan Cerebral's group in George Mason University. So we see here that the 3D DSA reconstruction, we see the computational fluid dynamics domain, and we can run a fluid dynamics simulation there using standard models for the blood and modeling this as a rigid wall. But then we can take this data set of this vasa vasorum, we can take the 3D model we made of the tissue specimen from the reconstructed micro-CT data set, and we can combine them together to have this computational domain for the transport study, where we're going to be modeling transport of oxygen coming from the vasa vasorum as well as from the lumen side, and trying to understand what happens when in the tissue and whether it's getting sufficient oxygen. So we can couple this fluid flow and oxygen coming from the lumen side with this transport in the wall. So this work then, as part of this study, we did two things. We looked at once without the vasa vasorum and once with the vasa vasorum. So the first case is saying, what if someone had, let's say, given angiogenic drugs, and so there was no vasa vasorum, or this body didn't develop a vasa vasorum, what would be the oxygen distribution? And the other is to say, here's the current situation. It has a vasa vasorum. What's the oxygen distribution? So we see on the inner surface, there's no problem, no surprise. There's blood in there that's well oxygenated. Oxygen from the blood is nicely diffusing into the tissue, and it's well oxygenated. On the outer wall, on the other hand, we can see that without the vasa vasorum, we have very low oxygen levels. There wasn't sufficient oxygen coming from the lumen side because there's cells inside which are using up the oxygen, which is part of the modeling, and so there was not sufficient oxygen at the outer wall. In contrast, we find that the vasa vasorum is able to provide levels of oxygen to most regions of the wall that's reasonable, even the outermost wall. And we can then look at cross-sections of this simulation to just look at how that gradation goes through the wall, because the model for transport is actually on the entire wall through the entire thickness. So we have data to see it's highest at the lumen side, and then in this case of vasa vasorum, there's also oxygen transport from the outer wall. So the main takeaways from this part of the talk are that we've developed this new 3D imaging approach that reveals a complex vasa vasorum network in brain aneurysms that's localized to the outer wall. It was previously unseen in 2D studies. And evidence suggests from this sample, and this is a new test that we've been doing, so we are now in the process of extending it to many more samples. Imaging for this one alone took 96 hours, so that's why we don't have many more samples here, and plus all the analysis. So from this sample, we can see that there's certainly a beneficial role for the vasa vasorum in providing tissue, oxygen to thicker walled regions and supporting this well-structured collagen and enabling effective oxygen delivery. So the clinical implications of this are we should have caution before targeting the vasa vasorum with drugs. We need to understand a little bit more about which cases the vasa vasorum is probably having a deleterious effect from those it's having a positive effect. For example, if we'd removed the vasa vasorum from this sample, we would have triggered hypoxia, which would have itself probably triggered inflammation wall degeneration. So further research is essential to understand this better and to have more understanding of this. Vasa vasorum are also very interesting because they're known to bring in a certain type of dye that can be imaged. And so there's a lot of interest in vasa vasorum right now because there might be another way of probing the aneurysm wall. So it's another motivation for studying this further. So I just want to end by just reminding us this is very much of a collaborative undertaking. I'm fortunate to have outstanding collaborators and to also have been well funded by the NIH. And I think it's important, just particularly at this moment in time, to emphasize that this funding from the NIH has been sustained and enabled sustained research, enabled us to have this team, outstanding team, brought together and kept together, and to be able to support graduate students and all of this imaging work. So I think it's very important to understand that even though the university is large, our research groups are really like small businesses. We cannot be disassembled and then miraculously go back together in 6 months. So this is something I feel very strongly about. And I also have a few seconds, I think, to say this is a cool thing. So here is another circle of Willis. And now here are circles of Willis from 2 animals. So you might wonder, like, what animal is that has a circle of Willis very similar to the human one? That's actually, well, you can think, but it's actually from a mouse. And this other one is from a sheep. It doesn't have this circle of Willis, it has a network. And there's a very interesting history of medicine investigations about the impact this had on science, because Galen, who was a big figure in the history of medicine, was using the sheep to demonstrate what human brains look like and convincing everyone, this is what human brains look like. And very few people were questioning that. And people who questioned it got in a lot of trouble. So maybe that's also something that's coming around again, unfortunately. But anyway, I'd just like to take this last moment to thank again the fellows and thank my research partners, undergraduate Harvard research partners, David Ahn and Evelyn Su. And also big thanks to the Harvard Radcliffe Institute. I am going to be eternally grateful for the opportunity to be here this year. It's had a huge impact on me and my outlook, and I'm very grateful to the dean. I'm also extremely grateful to Claudia and all the staff for always supporting us and making our life, work easily technically, but also just being very warm and friendly and supportive, and makes a huge difference. And in addition to this opportunity, it's also been a really great chance for me to reconnect with my friends who I haven't seen even from high school and from who live in the Boston area, and to make new, very, very dear friends. So I just want to end my talk by saying thank you, and looking forward to the discussion.
Thank you, Professor Ann Robertson. I have to say that again. It's interesting that we were about the same age, and at that time, engineering department was pretty much all male, sort of like a man studies program at that time. How did you get interested in engineering, and how could you achieve to be a successful scholar?
Well, it's nice to have someone from my high school here, because I remember a kind of pivotal moment when we were filling out some form about what we wanted to study in college. And I kind of liked biology, wasn't sure what to do, wasn't sure what to fill out. And I talked to somebody next to me, Leonard Greenberg, and he was, said he's going to write engineering. So I said, well, why are you writing engineering? He's like, well, I'm good at math, so I'm going to write engineering. And I thought, I'm better than him at math. So I said, maybe I should write engineering. And at some point, I started looking into it a little bit more and getting a lot of mentoring and things on that. So ended up going to engineering. And yeah, I was the first woman in my department, tenure track woman. There were not that many women. So there were a lot of unusual things going on. I think most of them were not poorly motivated, but they were impactful. But it's also very exciting to be able to make a difference so readily. I can have such a big impact on the women in my department, on the junior women coming through. And I think the hardest thing was finding mentors. Because a lot of people are just uncomfortable casually getting together with a younger woman to be a mentor. And so I had very, very few mentors. And so that's one of the motivations I had to start a mentoring program for junior faculty, which has been something which I'm very passionate about, and has done very well at the University of Pittsburgh. Every young faculty member has gotten tenure since we started this program for junior faculty. So it's very close to my heart.
That's great to know. Now also, given the frequency of these aneurysms in population, why don't we have population screening program nowadays?
Yeah, that's a good question. So, essentially, because the treatment has risks associated with it, if we started massively screening and finding all these aneurysms, then we would have to decide if we were going to treat them or not. And so it would even be a burden for people to know that they had these things and not have them treated. So the sort of consensus is, and also very expensive to treat all of these. So people who have like a familial history or have other high-risk things, they might be encouraged to go and have screening. But a lot of the aneurysms are found, only 10% have symptoms, so before they rupture. So they could enlarge enough to be pushing against a part of the brain where you have secondary effects, like trouble swallowing, like this woman had. So other than that, they can also be found incidentally. Someone was in a car accident, went in, had imaging done, so they can be found that way. But yes, it's unfortunate. That's why it's so important. It's one of the big questions if you ask neurosurgeons, what do you want us to do? What's the most important thing? It's really improved risk assessment.
I see.
Which ones are going to rupture.
Okay. And also, given the importance and it happens so commonly, and you get a lot of support from NIH.
Yeah.
Now, given the current situation, how will you continue to get support to treat this very important and common condition?
Yeah. So things are a little unclear at the moment, and I'm optimistic, but I was just looking on the NIH website, and currently in the institution, which I get my funding from, the funding rate they're expecting is around 8%. So 8% of all the applications that are submitted. It was already a very rigorous process where, I've been on both sides of that. I've been a standing member of the NIH study section for years, where I go to Washington, D.C. 2 to 3 times a year, and now people do it remotely, and sit at a large table with all these other scholars who analyze the applications and evaluate them. And it's a very, very rigorous process. So even then, the funding rate was 15 to 20%. So you had to be in that top level to get funded. So it's going to be tough. I know the NIH directors are really doing their best. So we'll see. I think it's important we all write in support of the NIH funding.
Yeah, wonderful. Yeah, we hope all the best.
Yeah, thank you.
There are quite a few these slide of questions. Here's a good one. How are aneurysms removed from a brain and transported to you? And I'm picturing a lunchbox.
Yeah, so we take that very seriously. So some of them are driven from Allegheny General Hospital in Pittsburgh, but most of them are transported through the mail. So very early on, we identified a FedEx, sorry, not the mail service, postal service. So we identified, we developed a whole kit. So the kit contains, the sample is put in a solution that's used for organ preservation, for transplantation. So a solution that's designed to keep tissue viable. And then it's controlled by temperature, it's cooled down. We have something that's recording the temperature while it's being transported, and we record that temperature. So we have a whole system and also, of course, we have to handle safety issues just in case the person had an illness that could be contagious. We have all sorts of precautions in case there's something leaked, there's a double bagging. It's a very elaborate process. But yes, it's not quite a lunchbox, but it's sort of a lunchbox on steroids, maybe.
But even it comes with a lunchbox, you won't eat it, right? Even you're hungry, yeah. Here's a technical question. Does the viscosity of the blood play a role in aneurysm and the ruptures?
Oh, okay. Yeah, so blood is a really fascinating fluid because it has plasma, which is sort of like water. It's not that complex a fluid, but then it has red blood cells in it, which are 40 to 45% by volume. And so they are very deformable and can flex around. And they can also form stacks. They kind of connect together if the flow is slow enough. And so there's a question of how do you model the viscosity? Is it a shear thickening fluid? Is it shear thinning fluid? In the body, so I've done some work in this direction, it becomes shear thickening, meaning shear thinning, meaning as you go down in shear rate, it'll look like it's thicker or more viscous. If you go up in shear rate, it looks like it's less viscous, so it's shear thinning. So that aspect is something that people have considered modeling and including in their models. But in fact, that physically, once again, looking into the material to see why that's happening because these red blood cells are forming these stacks, so-called rouleaux, three-dimensional rouleaux, which makes the fluid seem more viscous. But in order to get them to do that, you have to have it going really slow for a long time, like 10 minutes before they'll form. In the body, we have circulating through our body so rapidly, those are broken up so that doesn't happen. So the viscosity is relatively constant. Does it get thicker or more viscous inside the aneurysm? I mean, I suppose if we had cases where it was highly stagnant, these rouleaux could form, but our modeling is not pulling up cases that would have that level of stagnation.
I see, okay. There's a more technical question. Are aneurysms found incidentally? If not, can you say a little about the symptomatology of aneurysms and how it may be related to the physical and mechanical characteristics of aneurysm?
Yeah, that's a great question. Yeah, that's a very good question. Yeah. So where do the symptoms come from? So the symptoms can come from a microbleed, which can cause a tremendous headache. So a lot of times when this is just ruptured or about to rupture, people say they got the worst headache of their entire life. And that can be a symptom. There can also be symptoms from the aneurysm pushing up against tissue in the brain. And so that can show up in many different ways. That could have to do with, for example, with gait, with swallowing, with eyes, one drooping and things like that. So it's a very large range of symptoms. And so it's, yeah, it is complex. But yeah, certainly just like, any signs of stroke, because once it bleeds, it's a type of stroke, people should go immediately and get checked out as quickly as possible. And I think the other thing which I would advise is, if someone does have a ruptured aneurysm, try to make sure you're put in a hospital that has experts, neurosurgeons, who routinely treat aneurysms. My own experience with my mother is that, something happens, you get put in an ambulance and the ambulance driver says, where do you want to go? And, if you don't have a plan, that's where you go. And I know of people whose relatives had not gone to, get the appropriate care for a ruptured aneurysm. And that's unfortunate. That's very unfortunate.
Okay. One more question. Probably we have enough time. Are there any implications from this that can be reapplied to clots and the potential for a stroke, specifically for a large existing clot that is in a dangerous place to remove?
Okay. So, there's new technology that's being developed, certainly at UPMC in Pittsburgh that I know of, that I was helping with a little bit, where people can have clots lodged somewhere, forming, and now they try to actually go in endovascularly and attach something that can grab it as a claw or a kind of vacuum and pull it out. To do that, of course, blood, there's chemicals that can be given pharmacologically to try to dissolve these clots. But in terms of the aneurysm, it's really a different treatment modality. You're trying to remove something out of the body. Yeah.
So we have one more question.
Okay.
It's a shorter one, yeah. Did you ever consider working in medicine as a physician? That's a good one.
That's a good question. So yes, I did. In fact, when I started Cornell undergrad, I started as a double major studying biology to be possibly pre-med and studying engineering. I quickly realized my memory is not right for being a medical doctor. Just the capacity to memorize large amounts of information, that is definitely not one of my strengths. On the other hand, give me an equation, I can figure it all out and rederive it, whenever you need it. So I realized pretty quickly, this is the route I better go in, because this is where my talents lie. I have, a lot of respect for people who are medical doctors and who can retain all this information and make decisions based on that. Yeah.
Super. Thank you, Professor Ann Robertson, for your interesting presentation. And I also find that the images are beautiful, exquisitely beautiful, as well. I also want to thank our audience for their terrific questions. We have quite a few, and I hope you will all be able to join us for other Radcliffe visual programs. You can find out about the future programs and watch videos of past events at radcliffe.harvard.edu. Thank you again for joining us today. Have a good rest of the day.
Thank you.
Thank you.